Um, yes, so hello everyone. My name is Nikolay Oskolkov, and I'm a bioinformatician from Sweden, from an institution which is called SciLifeLab. It's a complicated abbreviation. It stands for Science for Life Laboratory, Sweden. And it's an institution in Sweden which does a lot of sequencing. So basically we sequence all possible types of all possible biological molecules, and we analyze this data. And I'm from the analysis platform of SciLifeLab, which is called NBIS, another complicated abbreviation. NBIS stands for National Bioinformatics Infrastructure Sweden. And we are supporting analytical projects from all over Sweden. We are a group of 70 guys spread all over Sweden, and we are doing support, bioinformatics support of projects from different academic institutions in Sweden. I just double check that, yes, so people are writing that screen and voice are okay. So meaning that, yes, so I can go to the full screen mode, and yeah, I will not check, I will not double check that, yeah, everything seems to be working. Yes, so the topic of today's webinar, yes, first of all, thank you very much for joining the webinar, and I really appreciate the invitation from RSG-Türkiye. So it's a great pleasure to give a webinar. Yeah, I'm going to talk about application of AI in biological and life sciences, and why biology and life sciences should lead the AI revolution. And we'll be talking also about data and big data for biology and life sciences, whether we have enough data in life sciences or whether it's still a bottleneck for our analysis. What you see on this picture, it's my dream. So I have a dream. I want natural and life sciences talk to each other, because currently they don't, in my opinion. They don't really understand each other, and they have dramatically different languages. So the situation, the connection between natural and life sciences is not as optimistic as on this picture, but rather like this. They look a little bit angry at each other because they have so different languages. And I know this for sure, because I'm actually from theoretical physics, and 2014 I switched from physics, from natural sciences to biomedical sciences and life sciences. And so I spent 10 years working as a professional physicist. I did my PhD in physics, did a few postdocs, so I was very deeply into physics. And then in 2012 I met medical doctors, real medical doctors who see patients, who do medical research, and they convinced me to switch from physics to biomedicine. And I spent 5 years doing medical research, doing diabetes and cancer research. And the last 4 years, when I switched from biomedicine to more, when I joined NBIS, basically, I've been doing more pure biology, pure biological projects, with a lot of evolutionary science, Darwin theory, stuff like this. And this is how I felt when I switched from physics to biological sciences. So I was completely lost in translation. And I discovered an effect which I call the white noise effect. White noise is just a random Gaussian signal without a noise, without an information. So just a pure noise. And when two biologists talk to each other, there was no information in their conversation for me. It was just a noise. So it was very, very hard to understand biological language. But then slowly I realized that there is a hope, and the hope is called bioinformatics. Why? Because bioinformatics connects people. Bioinformatics understands both mathematical language and biological language. And bioinformatics can translate between the languages. So I always recommend all PIs, if they have troubles interpreting or communicating between different research areas, they should call a bioinformatician, because bioinformaticians, they understand both languages. They're like bilingual children in this sense. So sorry for this very long introduction. It was just in order to explain that maybe sometimes you hear that I'm using strange language. It's because I'm from theoretical physics, so sometimes I will be using some terminology which might be new for you, and the explanation is because sort of I'm somewhere in between. I'm not a physicist, but I'm not a biologist. So I'm trying to combine the advantages from these different areas of research. So I was going actually to talk about big data for life sciences. And the natural question is, do we actually have big data in life sciences? Well, we can say what kind of question it is. Of course, we have a lot of data. Maybe you heard about DNA sequencing, huge, massive DNA sequencing efforts. Some people say that DNA sequencing amounts, they reach astronomical scales. So indeed, there are different public resources available, 1,000 genomes, TCG, EXAC. So the amount of sequencing in life sciences is increasing, and indeed reaches impressive scales. So it might seem like we have plenty of data for life sciences. And sometimes when people talk about big data, they use this common sense concept, common sense about what they really understand by big data. So I work on very different projects from life sciences, and often or every day I see PIs from different academic institutions in Sweden who say, you know, I have a lot of data and I want to run some machine learning, deep learning, artificial intelligence on this big data because it's really a lot of data. And then I'm asking, why do you think it's a big data? And the typical answer is that because it occupies a lot of disk space. So, for example, they say it's 500 terabytes data on my disk. That's big. Yes, it can be indeed a big data, but you know what? I can generate random Gaussian noise, one big file, which can occupy a lot of disk space, which can be 500 terabytes, but it will be still Gaussian noise. So it's not always useful to use common sense when we talk about big data, but rather what I understand by big data is advantages, computational and statistical advantages, which we can gain from analysis of big data compared to little data. So there should be some new mathematics arising from analysis of big data. But anyway, so if you look at genomics, whole genome DNA sequencing, it looks like it's a lot of data. TCGA, the Cancer Genome Atlas, massive amounts of data. UK Biobank, you probably heard that it's 500,000 of individuals with genetic information, with comprehensive phenotypic information. So there is a temptation to say that genomics is actually big data for life sciences. But you know what, since I was doing diabetes research a couple of years ago, I'm a big fan of this guy. His name is Mark McCarthy, and he's from the University of Oxford. And his lab is one of the world leading in genetics of type 2 diabetes. So Mark, last year, Mark was giving a series of talks at different places of the world. including SciLifeLab. And the title of his talk was, "Now we have found over 400 genetic signals influencing type 2 diabetes. What on earth are we going to do with them?" It doesn't sound very optimistic, does it? So why is Mark a little bit skeptic? Why can't we just take these huge genomics efforts and bring them to the clinics? This is what precision medicine is supposed to do. Precision medicine is supposed to use this huge knowledge accumulated from massive sequencing efforts, bring them to the clinics, and just take an individual DNA of a person and predict the inclination of this person to diseases or talents. So why can't we, in other words, why can't we predict the future from the DNA? Well, the simple answer is that it doesn't work like this. People tried and it doesn't work. And here there is a demonstration, a typical demonstration, why it doesn't work. So if you look at the plot at the right-hand side, this one. So here so this is a plot from a paper last year, September last year, from a paper in Cell. It was a paper about genomics efforts on abdominal aortic aneurysm patients from the group of Mike Snyder. And this group compared predictive power between genomics and electronic health records. And electronic health records, it's if you don't know, it's a blood pressure, it's a body temperature, it's height, weight. So it's quite a trivial information. And it turns out that this trivial information predicts abdominal aortic aneurysm so much better than genomics, which is quite frustrating because you would expect the opposite. So why is it like this? There are multiple problems here. There are multiple reasons why it is like this. One analytical problem, which I see, is that genomics works in ultra-high dimensional space. So if you're from Sweden, Swedish reference genome typically has 35 million genetic variants. So meaning that you're working in a 35 million dimensional space. Why is it a problem? Well, it's a problem because of the thing which is called the curse of dimensionality. What is it? So basically, when you do some statistical analysis on any type of data, you have two parameters, two dimensions. P, which is a number of features. Those can be genes, proteins, genetic variants, nucleotides, etc. And N. N is a number of statistical observations. It can be a number of samples, number of cells, nucleotides, etc. And depending on whether P is greater than N or N is greater than P, you can approximately select one of those three types of statistical analysis. You can run Bayesian analysis if you have very little data, if N is much less than P or P is much greater than N. If you have enough data, if you have N and P of the same order of magnitude, comparable, P is comparable with N, you can proceed with frequentist analysis. Whatever p-value based analysis you're doing, you're running frequentist statistics. Or if you have a lot of data, if you have a number of samples much greater than number of features, genes for example, you can still continue doing frequentist statistics, but you can do it so much better with nonlinear algorithms, such as artificial neural networks and deep learning. So now you can ask yourself, looking at your own data, where am I? Am I sitting here, here, or here? For example, in life sciences, it's very popular to run RNA sequencing. When you do bulk sequencing, you sequence transcriptomics from sick people maybe and healthy people from certain tissues. Can be blood, can be muscles, skeletal muscles, kidney, whatever tissue. So if you're talking about RNA sequencing, P is a number of features. This is a number of genes. And typically, if you're talking about humans, we have 20,000 coding proteins, coding sequences in our human genome. So P is 20,000. And N, so how many samples we usually have typically? What is the N in life sciences? It can be 100, maybe 1,000, maybe 2,000, but it's never 20,000, meaning that we are sitting here. So biomedicine is almost always sitting here, but it still wants to run frequency statistics. It's still, the biomedical analysis is heavily based on p-values, while actually we are violating assumptions of frequency statistics. If you are working in this limit, but want to run frequentist analysis, we are violating assumptions of frequency statistics. So what kind of assumptions I mean? So here I demonstrate sort of the essence of the curse of dimensionality. Let's take a simple frequentist type of analysis, which is a linear regression. So for example, it can be GWAS, genome-wide association analysis, or it can be differential gene expression analysis, or it can be differential methylation analysis. So essentially what you're running linear regression. So you have two variables, X and Y. X can be, for example, it's a matrix of genotypes from different individuals. And Y, this is a phenotypic variable. So it can be, for example, sick people and healthy people, diabetics, non-diabetics. So if you run this type of analysis, frequentist linear regression, this has a solution in a closed form. So you can actually express this slope beta in this form, in a closed form. And so here, what you have here, it's called inverse variance covariance matrix. So basically, in order to solve the linear regression problem, you have to calculate the inverse variance covariance matrix, which can be problematic. Because, so it's a complicated equation for the inverse variance covariance matrix, but you can think about that it's roughly proportional to 1 over determinant of the variance covariance matrix. Now, what if you are working in this limit, in a biological or biomedical limit, when the number of features is much greater than the number of samples? So if you go to high dimensions, then features or genes, they become correlated just by chance, because you don't have enough statistical observations to resolve the differences between genes. So once features are correlated, you know that genes are correlated, genes are co-expressed. Once they are correlated, determinant of such a matrix approaches 0, meaning that 1 over 0 goes to infinity. So the inverse variance covariance matrix diverges when you are working in this limit. So basically mathematics cannot stand this limit. Frequency statistics does not stand this limit. So by running frequentist type of analysis, p-value based analysis, when you have number of genes much greater than the number of samples, here you severely violate the very basic assumptions of frequency statistics. So does it mean that it's always dangerous to do biomedical research, that there is no hope? Because indeed, typical biomedical setup is here. We do have high dimensional data in biology and biomedicine. There is actually hope, and the hope is called single cell. In single cell, it becomes almost a routine to sequence thousands, hundreds of thousands, or even millions of single cell. One single cell is one statistical observation, meaning that you have millions of training examples for your analysis, millions of statistical observations for your frequentist or Bayesian or deep learning analysis. And this setup is a paradise for deep learning. So here you actually don't have to be concerned about overfitting. You know, in biology and biomedicine, we very often talk about overfitting, how to create a generalizable model which does not overfit, which predicts on an independent sample. But here, when you have millions of statistical observations, you have to watch out for underfitting. So it's a luxury problem. It's an upside down problem. And really, last year, for example, 10x Genomics, they published a data set with 1.3 million single cells. It's publicly available. Everyone can download and use it. There are works immediately after this study. There were papers published in 2 million single cells. And now there are resources which provide over 5 million cells publicly available. Everyone can use them. And this is a paradise for deep learning. Again, you can still run a p-value based analysis here, but you can do it so much better going outside of linearity, because frequency statistics is essentially a linear statistic. So when you have plenty of statistical observations, you can do a better fit going beyond linearities and doing non-linear type of analysis. And we use a lot of deep learning. So deep learning on artificial neural network, it's like the symbol of non-linear analysis. Deep learning is almost always non-linear, and it makes a lot of sense to use deep learning on single cell omics because of severe internal intrinsic non-linearity within the single cell omics data, because of some complicated reasons. As a rule of thumb, you can think that if the data has lots of zeros, if the data is sparse, then it means non-linearity. The sparser the data, the more non-linear is the data. That's why if you ask why deep learning works on image recognition or natural language processing, NLP, analyzing different texts, and what is common between images, texts, and single cells? Well, that's a sparsity. All three types of data are sparse. That's why it makes a lot of sense to run non-linear algorithms such as deep neural networks on single cell omics. And we use a lot of autoencoder analyzing single cell omics. We typically use autoencoders. Autoencoder is an unsupervised neural network. And why we want to do unsupervised analysis, unsupervised neural network? Because single cell analysis, if you're familiar with single cell analysis, you know that it's essentially an unsupervised analysis, because we usually use two unsupervised techniques when we do single cell analysis. Those are dimensionality reduction and clustering. They both are unsupervised. So here, this is a sort of here you can see a simple autoencoder. If you're not familiar with how autoencoders work, they basically try to reproduce their input at the output. So here at the output of neural network, you want to see, you want to reconstruct your input. This sounds like a little bit silly idea. Why would you want to try to reconstruct your input at the output? Well, it sounds silly until you realize that here in the middle you have a bottleneck. So autoencoders, they usually have this butterfly architecture. So there is a bottleneck in the middle, meaning that if you manage to reconstruct at your output, if you manage to reconstruct your input, passing through this bottleneck, it means that you were able to compress your data down to 2 dimensions only without losing information. And if you manage to do it, it means you learned something fundamental about your data. So you can really compress your high dimensional data down to 2 dimensions without losing much information. This is what autoencoder does. And how does it work in practice? So here I will demonstrate a typical application of autoencoder for single cell RNA sequencing data on 8,000 cells approximately for now. we will go to much bigger data sets in a few minutes. So, so here you can see a principal component analysis plot. So one point is one cell, and different colors here correspond to different cell populations. So you're supposed, those are blood cells, and you're supposed to see 12 cell populations, but you don't see them because it's a linear dimensionality reduction. So PCA, principal component analysis, is a linear dimensionality reduction. So it cannot resolve different types of cells. You can do it a little bit better with autoencoder, doing non-linear dimensionality reduction. So that's the huge difference. So it's not perfect. It's not, but you still see the 12 cell populations more clearly. You can do it even better if you use t-SNE, which is a golden standard in the single cell RNA sequencing area. But t-SNE has a bunch of disadvantages or limitations. For example, t-SNE cannot work on high-dimensional data directly. You always have to pre-process your data with, for example, principal component analysis. You always have to pre-reduce dimensions with PCA before proceeding to t-SNE. And it doesn't make much sense if you think that t-SNE is a linear. t-SNE is a non-linear dimensionality reduction technique, and PCA is a linear dimensionality reduction technique. So it doesn't make sense to do first linear dimensionality reduction in order to continue with a non-linear dimensionality reduction, because you lose some information. So you can do it much better if you pre-process your data with autoencoder. So basically, if you compare these two pictures, t-SNE on PCA and t-SNE on autoencoder, you can see that this purple cluster is more sharp here, because you basically constructed t-SNE using this picture compared to a situation on the left when you build a t-SNE plot on this kind of analysis. So here, when using autoencoders, you pre-processed your data so that it helps t-SNE to better resolve, to get a sharper clusters. So here, the data were pre-processed in a better non-linear way, which is easier for t-SNE compared to situation on the left when the data were pre-processed with principal component analysis. So I hope this makes sense to you. So autoencoders, they have a lot of potential for single cell RNA-seq or single cell omics area in life sciences. So you can say this was only 8,000 cells, so that's not a big data. So what about this 1.3 million cells? And here it is. So this is a 1.3 million mouse brain cells from the 10x genomics data set. And here again, you see principal component analysis. You cannot resolve all the cell populations. They are overlapping, heavily overlapping. You can do it a little bit better with autoencoder. You can do it even better with another beautiful nonlinear dimensionality reduction technique called Umap. Or you can use the old good t-SNE and good nice clustering as well. The problem with t-SNE is that it doesn't scale for this amount of data, for millions of cells. So basically, in order to obtain this picture, you'll have to wait for 1 week, which is sort of not feasible if you think about randomization and generating multiple images like this. So in this sense, Umap and autoencoders, they're taking over. So it's becoming increasingly more and more popular in single cell omics area to use autoencoders and Umap. So Umap and t-SNE, they have, in a sense, they're similar. They're both nonlinear dimensionality reduction techniques, but they also have some very deep and very important mathematical differences. So maybe, I don't know, maybe some of you don't know what t-SNE is, but then maybe you don't have to know this. Because as I said, t-SNE is basically, it has some limitations which are not really resolvable, and we can the single cell community switches from t-SNE to Umap more and more. So to explain the difference between t-SNE and Umap, you have to sort of dig into mathematics. So if you happen to be familiar with how t-SNE works, you know that it's based on basically the following 4 equations. What t-SNE does, it makes a distribution of distances of points in high dimensions as similar as possible to the distribution of distances between points in a low dimensional space by using this cost function, which is called the Kalbeck-Leibler divergence. So here P is a probability to find points at a certain distance in high dimensions, and Q is a probability to find points at a certain distance in low dimensions. And t-SNE tries to collapse, to project high dimensional probabilities into low dimensional probabilities. This is how t-SNE does dimensionality reduction. So the cost function is the main difference between t-SNE and Umap. So if you now look at the Umap cost function, which is called cross entropy, so it looks pretty similar to t-SNE, but here there is one more term. And this term, it makes dramatic differences in the final output. So this term exactly, this is what makes Umap to be able to preserve global distances and not only local distances. So if you're familiar with t-SNE, you know that t-SNE guarantees preservation only local distances. I will explain in a couple of seconds what it means. While Umap can preserve both local and global distances. So Umap is more suitable for clustering, if you want to cluster on Umap representation. So now, in order to deeper understand what I mean by local distances and global distances, we have to plot t-SNE cost function, which is Kalbeck-Leibler, and Umap cost function, which is cross entropy, as a function of x and y. Here, x variable, it's a distances between points in high dimensional space, while y, it's a distances between points in a low dimensional space. Now let's take points which are far apart in high dimensional space. So x is large. So now we are looking at the Kalbeck-Leibler plot. If the distances between points in high dimensions are large, then in sense of Kalbeck-Leibler divergence, you really want to have, sorry, they are small. Here it's actually, I'm pointing here at small x. So when the distances between points in high dimensions are small, then you really want to have y equally small in order to have as little penalty as possible. So the Kalbeck-Leibler divergence will be low. So Kalbeck-Leibler is sort of a penalty. You want to minimize Kalbeck-Leibler. So meaning that if you have points which are close to each other in high dimensions, t-SNE guarantees that they will be close to each other in low dimensions as well. However, if you consider points which are far apart from each other in high dimensions here, so if you take large values of x, then in sense of Kalbeck-Leibler divergence, there is not so much difference here. So you can have large y, or you can have low values of y, and there is not so much difference in penalty. Meaning that if points were far apart in high dimensions, t-SNE does not guarantee that they will be far apart in low dimensions. They can equally well happen to be close to each other in low dimensions. But look at the cross entropy cost function. So here, if you consider this limit of large x, here you really want to have a large y as well in order to have as small penalty, as small cross entropy penalty as possible. Because if x is not large but small, you get a huge penalty here. So meaning that Umap ensures that the points which were far apart in high dimensions, they will be far apart in low dimensions. That's why the distances between clusters in single cell omics, they make sense if you do it with Umap. But they do not necessarily make sense if you do it with t-SNE. This is what I mean by global distance preservation by Umap and not by t-SNE. So here, this was just a brief explanation of the major differences between Umap and t-SNE. So here I wanted to emphasize another major application of deep learning for single cell omics. This is a Bayesian deep learning. Bayesian deep learning is so it became very popular a few years ago in the area of self-driving cars. Basically, when people realized that in order to make safe predictions about how the car is supposed to drive by itself, so the safety apparently is a big issue for self-driving cars. And then people realized that regular frequentist deep learning was not good enough. And there were a number of attempts to make a safer predictions, safer modeling, introducing Bayesian framework. And for us, for people doing life sciences or biology-related sciences, computational biology, if you think about biomedical applications, the safety is also an issue for biomedical applications, because the price of misdiagnose, the price of making a wrong prescription and wrong diagnose is high for biomedical research. So we are not allowed to be mistaken in our prescription. That's why frequentist neural networks and frequentist type of analysis does not give you safe enough predictions, and people switch currently more and more towards Bayesian type of prediction in biomedicine. So here I'm just demonstrating how it works for single cell. So this is a t-SNE plot of cancer-associated fibroblasts. So different colors here mean different cell types. And here you see predictions of neural network. So the neural network tries to discriminate between different cell types, and it was trained on a much bigger data set. So here you see how neural network predicts on a test data set. And these are the borders it draws. So the predictions of the Bayesian, or in this case, it's actually a frequentist neural network here on this plot. The Bayesian, the predictions are based on these boundaries between different cell types. So basically, what you see here is that look at these two yellow guys. They fall onto a red background, meaning that the neural network was here 100% sure. So the red background means that it's 100% probability that those two yellow guys, they actually belong to the purple cluster, which is a severe misclassification. So those are actually yellow cell types. But the neural network, the frequentist neural network, predicts that they belong to the purple cell cluster. Bayesian deep learning can give you also uncertainties. So it besides this heat map, it generates another heat map, which is uncertainty heat map. So frequentist neural network can only provide you this type of picture, but not the other one. So the other one, it's a peculiarity of Bayesian analysis. So here, you can see that those two yellow guys, they fall onto somewhat darker area, meaning that the neural network, the Bayesian neural network in this case, was not at all sure about these yellow cells, whether they belong to the yellow cluster or to a purple cluster. So indeed, uncertainties, it's another level of information which you can incorporate. So basically, in this case, the Bayesian neural network would say, I'm not sure how to classify those two yellow cells. I need more information. While frequentist neural network would say, I'm 100% sure that these two yellow cells, they're actually purple cells, which is a misclassification. So in other words, put it simply, if you train your frequentist neural network to recognize cats and dogs, what would it say if you show it a picture of a bird? Well, the frequentist neural network would say, well, it's a cat, for example, because how can it know that it's not a cat? It was trained to recognize cats and dogs. It's never seen birds. How can it say that it's a bird? But you know what? Bayesian neural networks, they can make miracles happen. What Bayesian neural network would say, it would say, well, it's a cat, but with a very, very high uncertainty. Meaning that Bayesian neural network would say, I cannot safely predict whether it's a cat or dog. And this can be important if you make if you're not allowed to make mistakes, if you're not allowed to misdiagnose patients, if it's about safety. So if you don't believe me that Bayesian neural networks make this kind of predictions, you can make a simple experiment. You can take a typical benchmark from machine learning, which is called fashion MNIST, which is a set of images of different clothes items. It can be dresses, t-shirts, trousers. You can train a frequentist neural network to recognize those images. So it's an image recognition problem. And now you want to make a prediction. You take a picture of pullover, which was from a test subset. So the neural network has not seen this particular pullover picture. And now you want to make a prediction on this picture. And the neural network says, yes, it's a pullover, 100% sure. So this y-axis shows the probability of prediction, 100% sure. But what happens if you take a picture of a car? Well, the neural network says it's a bag. And this is not surprising because the neural network has not seen cars. It's seen only clothes items. How can it know that this is not a bag? Well, indeed, it might look like a little bit like a bag. But you know what? Again, Bayesian neural networks, they can make miracles happen. Let's now train a Bayesian neural network to recognize these fashion MNIST clothes items. And now we make predictions. So again, we take the picture of pullover, and the Bayesian neural network says, yes, 100% sure that this is a pullover. And now we take a picture of the car. And now the Bayesian neural network says, well, it's a bag, 50% probability, but it can be also a trouser, 50% probability. So you see that the Bayesian neural network was not at all sure whether it's a bag or a trouser. So this means that the Bayesian neural network basically says there is something wrong with the data. I cannot make a safe prediction. I need more information to make a right conclusion. So this is where Bayesian neural networks can make a dramatic difference when it's about safety of predictions. So here I'm talking mostly about single cell omics in this talk. And we use a lot of deep learning for single cell multi-omics integration. So data integration, it's another important and growing area in life sciences. When you have multiple sources of information from biological cells, such as DNA, RNA, protein, and you want to basically combine those types of information to better model behavior of biological cells. And there are actually technologies which deliver multiple types of information like transcriptomics, DNA methylation, and ataxic chromatin accessibility maps from the same cells. And to do this data integration type of analysis, there are quite a few methods developed in this area. They do not necessarily scale for millions of examples, and most of them are very linear. One non-linear type of analysis would be to do, again, neural networks for data integration. By the way, different people mean different things by data integration. So what exactly I mean by data integration? So here I demonstrate what I mean by data integration. Think you have two omics. It can be RNA sequencing and maybe methylation. And you have maybe 20 samples. So they're shown here, 20 samples. And those 20 samples, they belong to 3 different classes. Maybe diabetics, pre-diabetics, and non-diabetics. So if you... So try now, so what the axis here mean, the x-axis and y-axis, those are some latent variables which capture information from this omics. Like, for example, if you take principal component analysis, then the x-axis can be PC, principal component 1 from RNA sequencing, and y-axis, it can be PC1, principal component 1 from DNA methylation information. So now try to project those points, those data points at the x-axis. You will see that you cannot really discriminate between the 3 classes, the black, red, and green classes, based on a single omic, because those data points, they overlap. You cannot separate the 3 classes if you use only one omic. The same is valid for the y-axis. So if you project those points at the y-axis, they will be heavily overlapping. The different colors will be overlapping, meaning that you cannot safely discriminate between different classes. But if you plot those 2 omics against each other, you can see that in case the 2 omics co-vary, you can now draw linear boundaries. You can just use 2 planes to separate, not perfectly, but pretty well. You can separate the black points, red points, and green points. So this is the beauty of data integration, to see patterns which were invisible in individual omics. So individual omics, RNA sequencing and DNA methylation were not enough to discriminate between sick and healthy people. While when you put these 2 omics together, there is a mean, there is a way to that these 2 omics, they reinforce each other. There are some synergy between them, and these synergetic effects, they allow to better classify healthy and sick people. And we do a lot of omics integration again with autoencoder. And here I demonstrate how it works in practice. So, for example, if you have single cell RNA-seq data and single cell proteomics data, those are t-SNE plots from 2 individual omics. What happens if you combine these 2 types of data? This is the resultant image. And here you can see that once you combined RNA sequencing and proteomics data, you can see that this cluster becomes more visible now, the purple cluster. While if you used only single-cell RNA-seq data, you would not be able to separate the purple cells from the blue cells. If you run clustering algorithm, the clustering algorithm cannot really draw this boundary because the purple cells, they overlap with the blue cells. Well, now almost any clustering algorithm would say that this is a separate cluster. So basically, combining different types of information, you discovered new biology, you discovered something new. This is a beauty of data integration. So I hope I delivered my main message that single-cell omics represent a truly big data for life sciences and computational biology. So this is a big data number 1. single cell. Big data number 2 in life sciences, it's microscopy imaging. That's not surprising. You probably heard that there are automated microscopes, automated microscopy, which generates hundreds of thousands and millions of images. And they are those amounts of images. They are ideally suited for deep learning because of different reasons. Because it's a huge number of samples. It makes a lot of sense to do nonlinear analysis, and also deep learning, convolutional neural networks. They can do automatic feature extraction, so you can actually feed the raw image pixels into a neural network. This is very different compared to traditional machine learning based segmentation of images. So people did image analysis previously with machine learning, doing manual segmentation, so basically emphasizing that and showing the algorithm where the features are, while deep learning is capable of detecting features. Features can be cells, for example, by itself. So you don't need to say that here there is cell number 1 and here there is a cell number 2. So there are public resources, like in Sweden we have the Human Protein Atlas, a public resource with a lot of biomedical microscopy imaging, and you can do different types of image analysis. You can do image classification. It's a standard type of analysis, like discriminate between good cells and bad cells. Or you can do object detection type of analysis. That's another type of neural network. It's not a traditional convolutional neural networks, but so-called regional convolutional neural networks, which can do, it's sort of a combination of two neural networks. One neural network tries to detect object on an image, like here. You can see, so here my problem was to detect cells with a protein expressed in nucleoli within cell. And one neural network tries to detect those cells, and the other neural network tries to classify those cells. So that here the neural network correctly predicted that those two cells, they have a certain protein expressed in nucleoli, while other cells they don't. So it's another type of problem, object detection. So the third big data in life sciences is actually genomics. So you remember I said that, yes, we have a lot of genomics data, but we cannot really do not only nonlinear analysis, but even linear analysis would have troubles because of the curse of dimensionality. That's correct if you consider one genome as your statistical observation. However, what if you consider one sequence from a human genome as a statistical observation? Or in other words, you can chop your genome into multiple sequences, and those represent a truly big data. So there is a typical misunderstanding between biomedical people and people who do evolutionary research. For example, analyzing Neanderthal DNA. Biomedical people say, you have only one Neanderthal sequence. So what kind of analysis you can do with n equal to 1? Well, the misunderstanding is because biomedical people are used to working with tens of thousands individuals, hundreds of thousands single cell data, but they don't realize that one genome is actually big data if you chop it into multiple pieces. So indeed, if you consider one sequence or read in next generation sequencing terminology as a statistical observation, then one genome is a big data. So here I just quickly demonstrate how you can use deep learning or convolutional neural networks for very exciting type of genomics, which is ancient genomics or ancient DNA. So you actually can, so for ancient DNA, if you're not familiar, ancient DNA is very fragmented because of some strange chemical processes. So with time it gets more and more fragmented. And modern contamination, it's a big problem for ancient DNA, because if I drop my modern DNA into an ancient sample, there is no way I can see just by looking at a sequence whether it came from Neanderthal or from me. So there is no way to just visually discriminate between ancient and modern sequences. So we humans cannot do it, but neural networks can. So you can really train a convolutional neural network to discriminate between ancient and modern sequences. You can achieve good accuracies of prediction. And if you try to interpret the neural network or create so-called saliency map, you try to understand the importances of each nucleotide in a sequence. So where does the signal come from on a sequence? Does it come from the middle of the sequence, from the left part of the sequence, from the right part? And here you can see that the signal comes from the ends of the reads of the sequences. Meaning that the ends of the reads, they are most informative, most predictive, if you want to assign an ancient status, if you want to classify whether it's an ancient DNA or non-ancient. And this makes a lot of sense because actually there is a process which is called deamination or damage pattern. There is a concept of damage pattern. It means that ancient DNA degrades from the ends. So meaning that indeed the ends of the reads, they are very predictive in sense of ancient status classification. So I hope I delivered my main message that we do have big data in life sciences. We have single cell, we have microscopy imaging, we have genomics, if you consider one sequence as a statistical observation and not one genome as statistical observation. So there is plenty of work to do in life sciences, and those amounts of data, they are indeed perfectly suited to be addressed with nonlinear algorithms such as deep neural networks. So here I stop. I am from the National Bioinformatics Infrastructure, Sweden. And I am financed by different sources, Knut and Alice Wallenberg, Vetenskapsrådet, and physically I'm from Lund in Sweden, Lund University, employed by Lund University. So I thank you guys for your attention, and it's been 1 hour, so I'm on time. Thanks.
So thank you for your really informative webinar. It was not really nice summary for deep learning applications for the life sciences. If you have any questions, you can ask right now from the chat section at the right-hand side. And by the way, sorry about the problems at the beginning of this webinar. I had some connection issues. Sorry about that. If you have any questions,
so please just ask me. Just type your question, or maybe I can hear you. I don't know. At least I see what you guys are writing.
Meanwhile, I can ask one question about single cell analysis. You mentioned that t-SNE is replaced by Umap, and recently I've started to see some other dimension reduction techniques for other purposes. Do you think that those different dimension reduction techniques should be considered, or what do you think about the other developed techniques about dimension reduction?
Yeah, so if you're talking about single cell, I think, as I said, people are switching from t-SNE to Umap and autoencoders. The advantage of Umap is that it's very easy to run it. It's very fast. It doesn't need much resources. It doesn't need much memory. It doesn't need a powerful computer. You can run it on your laptop, even on millions of cells. Autoencoders are difficult to train. They are not that straightforward to implement, but still they have a great potential, because autoencoders, neural networks. So neural network is a perfect computational umbrella which can mimic any other computational method. So basically, to my understanding, if you want to come up with your own dimensionality reduction method, it's possible. Usually they are based, I think Leland McInnes, the developer or inventor of Umap, he brilliantly emphasizes in his lectures on YouTube that there are basically two types of dimensionality reduction methods, which are matrix factorization and neighborhood graphs. And t-SNE and Umap, they belong to the second class, to the neighborhood graph. So you can definitely develop many types of neighborhood graph type of dimensionality reduction just by changing the cost function. So if you come up with a smart cost function, what you minimize, what you optimize doing the dimensionality reduction, you definitely can come up with a dimensional reduction which suits better your particular type of data. So Umap and t-SNE and autoencoders, they work very well on single cell data. They were optimized. They were tested a lot on single cell omics data. If you're working with texts or images or maybe some data from astronomy or econometrics, you might use some other different dimensionality reduction techniques. But I think essentially, like conceptually, what you're doing, you're either doing matrix factorization on neighborhood graph, and you slightly tune cost function, which is more appropriate for your type of data. Yeah, so that would be my comment. So I can see a question. I wonder if, yeah. So Batuhan, sorry, did I manage to answer your question?
No. That's okay. My question was that. And Handan asks, is there any uniform ranking method for dimension reduction problems? And Umap separates in global distances better than t-SNE. Do we say it by looking at the results by eyes? What do you think?
Yeah, the peculiarity of Umap is that it's safe actually to run clustering on the 2D Umap image. So with t-SNE, the difficulty about t-SNE was that you do dimensionality reduction, you visually see clusters, but it's not okay actually mathematically. It's not okay to run clustering algorithms on the 2D image, because t-SNE does not guarantee that the distances between clusters, they are meaningful. For example, hierarchical clustering is impossible to build on the top of t-SNE, because hierarchical clustering, it takes into account relations between different clusters, which clusters are closer to each other. But it makes more sense, so it's more okay mathematically to run hierarchical clustering if you want. There are different types of clustering, so I would recommend density-based clustering or graph-based clustering, and you should be perfectly fine to run it on the 2D Umap image. And in addition, if you're familiar with t-SNE, you know that t-SNE basically can do dimensionality reduction down to 2 or 3 dimensions only. So you cannot create, or it's not straightforward to build 10 dimensions in t-SNE. if you want it for general machine learning purposes, for prediction purposes. Well, in Umap, you can do it. You can do dimensionality reduction down to 2 dimensions, or down to 3 or 5 or 10. So here you do not have a limitation. Computationally, it's not limited. While computational issues were actually the limitation. Why t-SNE could not do dimensionality reduction down to 2 or 3 more than 2 or 3 dimensions. Yeah, I hope that answers your question.
And another question from Emre. He wonders if you had encountered with any particular algorithm design for bioinformatics like saliency map.
Yes, a major critics for deep learning applications is about not clearly understand the system. So people are trying to answer why questions. Did you encounter with any particular algorithm design for bioinformatics like saliency map? I mean, so there is a common misconcept that deep learning and neural networks, it's a black box, and they have problems with interpretability. I think it's not fully true, because as I demonstrated, for example, for ancient genomics, that you can really extract feature importances. You can really understand from what nucleotides the signal comes. And if you do, I don't know, if you run deep learning on single cells, you still can extract gene importances. You can understand which genes drive separation between clusters. So any machine learning, not only deep learning, but any machine learning technique will give you 2 things. First, it's a trained model which can predict. You can take this model and go to the clinics in order to advise medical doctors, for example. The second thing is that any machine learning delivers feature importances. So any machine learning will give you which features drive your prediction. So if you know what features or what genes are responsible for the prediction, you know pretty much about your system. You can deeper. You can understand biological processes much deeper, because you know the drivers of your biological pathway, the key players in your biological process. So you still can interpret to a large extent what is going on, why your model is capable of making such good predictions. So I do not think that actually deep learning is a black box. Yeah, so saliency map, it's a way to saliency map or GradCam for image recognition. It's actually a way to interpret neural networks, and there are actually multiple ways to do it. So it's definitely not a black box.
Do you have any other questions? If you don't have any questions, we can end the webinar. So I would like to thank again for your webinar. It was really nice and informative, and thanks for the participants for their support. Our next events will be announced on BigMarker, Facebook, LinkedIn, and Twitter accounts. You can register Bioinformatics, our webinar channel, and if you register, you can have notifications about our new webinars. So thank you again, and have a nice day.
Thank you very much. Thank you very much. Have a nice day and good luck with your analysis.